All right, I think we can get started. If most people are back in the room, are they? I think so. So I think what you heard from Rudy in the first lecture was kind of some high-level concepts in terms of why you would want to generate this kind of data, what the quality of the data is, and how you might, how you would use that biologically. And I guess my role here is to say something a little bit more about how you generate this data, so the details of how data DIA works at a low level, because we're going to be looking at a lot of this data tomorrow in the tutorials, so it's worth knowing something about how the data is generated. So I have two talks, one now and one in the morning, I think. But I'm going to split this into basically three sections. So I hope what's going to happen is, first we're going to talk about introduction to DIA, so how do we operate the machine, what are the basic concepts. Second, and I guess I might get through sort of half of this part today, we'll talk a bit about spectral libraries and prior knowledge and why it's useful to know something about about the peptides you're looking for, so why it's useful to know something about how the peptides look to the mass spectrometer before you try to extract them from this highly kind of complex DIA data. And then part three, which I guess from in the scope of this course is going to be very important, is some of the data analysis concepts and the kind of low-level details about how we do the data analysis, about the signal processing, about scoring the data, about false discovery rate control, as Rudy mentioned in his talk. So that's going to come tomorrow. So I hope this sort of splitting this one across two talks is not too incoherent, but I thought it was kind of more useful to keep this high-level structure of these three lectures separate. Okay. One thing I want to mention actually is that we run a DIA course in Zurich, and we usually record all the lectures from these videos, so if you're interested in getting more information, you can go to this YouTube channel, this targeted proteomics course. And until now, for the last sort of 2-3 years, I think there's 3 years worth of videos on there now. There's been about 50,000 views, so I don't know who's clicking on these things, but somebody, maybe it's bots or I'm not sure, but hands up anybody who actually looked at one of these videos. Anybody? Yeah, a few people. Okay, so real people. All right, that's good. Okay, so here's the outline for the first part. So first, a little bit about why do we need DIA. This is a little bit redundant with what Rudy said, so we can kind of skim over this. But more importantly, what do we actually mean by data independent acquisition? So how is the instrument actually operated in this mode? What are the kind of principles of operation of DIA? And then something about how we would start to analyze the data. So in what we have called in the past targeted data extraction, or peptide-centric analysis is the term that's mostly used in the McCoss lab. What some advantages of having this type of data might be, and Rudy also touched a bit on this, but I'll show some other specific examples that might be helpful in illuminating what some of the advantages of this type of data might be over other types of proteomics data. A little bit about kind of current status, so what can be done, and actually this is a little bit out of date, but I think it showed a little bit of data from a cross-lab comparison study that we did that I hope illustrates some of the potential of DIA and maybe a little bit of outlook. So firstly, why do we need something other than shotgun or DDA or more classical targeted proteomics approaches like SRM or pre-RM to quantify proteomes? So Rudy already showed some pictures like this, but let's just to state the obvious. This is let's assume that the proximal goal of any of these experiments is some kind of quantitative matrix where we have some set of samples or conditions on one axis and some set of peptides or proteins on the other axis, and this, can be arbitrarily large. And Rudy mentioned that it might be useful to have it larger in one dimension in this dimension than the other. But what we would like is a nice complete data set here so we can do the types of analysis that he discussed. So this is the goal. And he also showed basically some version of this slide that says, well, the one on the top here is what the data looks like. Well, let's start with this one. This is what the data looks like from DDA, at least maybe not now, but historically. The DDA data, you could get very deep coverage, but it took a long time to acquire the data, and the data was full of holes. We had this missing value problem. This pushed people in the direction of SRM and PRM, where you could now measure large numbers of samples faster, and you could get much more complete data but it was restricted to a discrete set of proteins, which is on the order of 100 or 200 proteins that you know the identity of before you start. So what we would like, is this quality of data, but we would like to be less restricted in terms of how many proteins we can measure. So we want to maintain this quantitative data quality, but essentially go deeper and not be constrained with having to know in advance what we want to measure. Okay, so I'm going to show a bunch of 3D projections of the data just to illustrate how the precursor space is sampled in various mass spectrometry methods, first in DDA and SRM. So for many of you, I think this is going to be revision, but I think it can be helpful just to mention this again, and then we'll look later on what it's going to look like in DIA. And what the projections here are these sort of 3 dimensions that we're always talking about in mass spectrometry. So we have the mass to charge dimension here, we have the intensity dimension, and we have some chromatographic dimensions, so retention time. And if you look from this projection, you're going to see a mass spectrum, so it'll be M over Z versus intensity. If you look from this projection, it's going to be the chromatographic projection, so you will see chromatography, like extracted ion chromatography, for example, but you can also project this in 3 dimensions. So let's see how it looks for DDA. So in DDA, what we have now is a zoom in of the mass range. We're zooming in from 500 to 525. And what you can see, what's happening is that each one of these gray lines is a precursor scan and MS1 scan in the instrument. And you can see that there's some peptides eluting chromatographically, so they have this nice chromatographic elution. And we have a blue one and a red one and a couple of gray ones. And each precursor is sort of a snapshot. Each MS1 scan is a snapshot of the precursor space at a given time in the chromatographic elution. And in DDA, and so of course we can extract actually quantitative values in the MS1 space by doing extracted ion chromatograms, and that's what's indicated here by these lines. And in DDA, the way the machine is being operated is that the instrument says at each point in time, for each precursor, for each MS1 scan, it says, well, what set of peptides are present, and it's going to make a decision on the fly about which peptide it wants to select for isolation and fragmentation. And that's what's represented by these boxes here is the precursor isolation space, right? So the precursor isolation window, I should say. And so in this case, this gray one is being selected in this first scan, and we're acquiring an MS spectrum where this gray peptide has been selected, and we have not a very informative MS spectrum here. Then the blue one comes up, and we're isolating this blue peptide, and now we have a bit more informative looking MS spectrum here, and then the red one and the gray one and so on. And the instrument just continues to do this in a sort of in a data-driven way, data-dependent way for the rest of the run. And so the features to notice about this is that each MS2 spectrum is a snapshot of a different peptide, so it's pretty fast. You can get a high number of identifications. It's instrument-driven, and it's going to be biased towards the high-abundance species, and it's not going to be consistent across samples because there's some kind of stochastic behavior in terms of what the instrument is seeing from one precursor scan to the next, and it's essentially not going to behave in a deterministic way. And so the important thing to notice here is that this precursor space is being sampled by MS2 spectra discontinuously in the mass and time dimension, right? You have sparse coverage of the precursor space with MS2 spectra. So this is in contrast now to SRM, where we have now the same set of peptides, but here we're just going to instruct the instrument to acquire deterministically spectra or signals rather for a set of peptides that we're particularly interested in. So we know that we're interested in this blue and red one, and here again these boxes are indicating the precursor isolation window of the quadrupole, and it just deterministically acquires, isolates this particular red or blue peptide, and we collect signals then in the Q3 space, in the fragmentine space that correspond to fragmentines that we're aware of previously. And so we can then extract quantitative data, and those of you who are in the earlier module focusing on Skyline will have seen a lot of pictures essentially like this one, where we have ion chromatography that we can use to determine that this is the peptide that we're interested in and eventually quantify it. But the important thing to notice here is essentially that this precursor space which we're talking about with these boxes, is sampled by MS2 signals continuously in the retention time dimension. So we have very nice deterministic coverage in the retention time dimension, but we don't have complete coverage. We don't have continuous coverage in the mass to charge dimension in terms of the precursor space. So there's a whole bunch of things. There's a whole bunch of this precursor space which is missing. And essentially, each MS2 series is a recording of one peptide across the LC, and the fragmentines are acquired over time and used to reconstitute the elution profile. So this can be very consistent because it's done in a deterministic way. There shouldn't be missing values, and this drives accurate quantification. So it's entirely driven by the user or by the machine. The problem is that we have this low number of precursors monitored per run. So I guess this should be reasonably well understood by everybody. Is that new to anybody, or is this basically understood? Okay. So, and the goal in terms of SWATH or DIA was to expand this. We wanted to get the data quality of SRM shown in this previous heat map, but maintain the number of precursors that we could monitor. And this was done by combining basically two things: data independent acquisition, where we use wide precursor isolation windows to deterministically or to continuously cover the precursor space, plus prior knowledge. And the idea was that by combining these two things together, we could get back to something which is SRM-like quantitative data, essentially. And so the goal was to go from this type of matrix, which is the SRM type of matrix, to this type of matrix, which is hopefully what the DIA matrix should look like, and what you've seen in Rudy's talk already. Okay, so just to make a few comments about what we mean about data independent acquisition. So this DIA term was introduced in this paper 2004 from John Yates, and it's essentially, I mean, the first thing to say is it's explicitly not data dependent acquisition, right? So there's no stochastic precursor selection going on, and the features of DIA are basically that the duty cycle is entirely fixed, right? So it's entirely deterministic, and this is kind of in a sense like SRM or PRM, but unlike DDA. However, it's also untargeted and unbiased data acquisition, so we don't have to provide a list in advance of targets. And this is in a way like DDA, but unlike SRM or PRM. And what we have, hopefully, what we're trying to achieve is comprehensive sampling of the precursor space. And usually this requires wide precursor isolation windows, and this usually means you end up with very complex mixture MS2 spectra. Okay, so, and Rudy and Brendan already brought up this nomenclature issue. So there's a whole bunch of different kinds of DIA which have been published over the years. So this is a kind of an attempt to summarize this in a review, actually a tutorial paper written by Tina Ludwig that's in revision at the moment. We hope will come out soon. But essentially this is all these different kinds of different names for different flavors of DIA that have been published since around 2003, I guess. And they're organized by time, so the earliest ones at the bottom up to the most current ones. And they're also organized by width of the precursor isolation windows. So we have the lowest width precursor isolation windows is Pacific over here from Dave Goodlet's lab. We are usually operating in SWATH in terms of this kind of medium width isolation windows, all the way over to where you don't really have any isolation width at all. This is shotgun CAD or MSE. So this is just as a kind of a reference to say, well, there are many different flavors. They share some common characteristics in terms of being data independent acquisition. And the development, I think it's interesting to look at how the development has gone on. So this is now a PubMed search I did actually 1 or 2 years ago, so I should probably update this slide. But this is a PubMed query which is trying to capture the set of papers which are using DIA or SWATH in the realm of kind of proteomics. And you can see that there was this phase from around 2000, early 2000s up until kind of around 2012, where people were publishing here and there 1 or 2 technical or proof of concept studies, but it didn't really take off until around 5 or 6 years ago. And what happened there, I think, was that a couple of things happened. One is that the instruments got faster, but also the approaches to the data analysis, I think, became a lot smarter. And I'll say what I mean by this over the course of the next 2 lectures. So what's been happening in this phase has been much more intense method development, but eventually as we go up here, lots of practical implementations, and now people doing a lot of applications. And I think actually even more so in the last year, it seems like more people are really adopting this. One other thing to point out is that the type of instrumentation that's typically being used here for DIA is usually some combination of a quadrupole, mass selective quadrupole, and a high resolution mass analyzer. So this is a time of flight, or in this case, an Orbitrap. So that doesn't mean that other configurations can't be used. They probably can, and I think this will be an interesting area for further research. But at the moment, the most popular applications are on one of these two configurations, and increasingly the Orbitrap type of configurations. Okay, so let me get back to this kind of three-dimensional projections. So now we talked about how SRM and DDA are working, and we sort of roughly sketched out what are the features of DIA. So how does it work in this same projection? So essentially what we're talking about is now we are increasing the size of this precursor isolation window. So this red box again is indicating the precursor isolation window. And what that allows us to do is capture all of these peptides and collect MS2 fragmentation spectra for all of them effectively together, right? So what you get is MS2 scans, which are very highly complex and contain fragmentines from all of these four peptides, which are in the precursor isolation window. So you get a kind of a zoo of data here, but the one interesting feature is that the MS signals, or the sampling of the precursor space, is continuous in the mass dimension and in the retention time dimension. So the retention time dimension is obvious, but if you stack a bunch of these windows together, you put another one of these precursor isolation windows here, you can eventually cover the whole mass range, and you have essentially both deterministic and complete data. But it comes at the cost of having these very highly complex MS spectra. And so you can see that in these blue and red peptides, this information is contained in there, but it's in the background of a whole bunch of other things. However, if you introduce some prior knowledge, you say, I already know something about what this blue peptide looks like to the mass spectrometer, I know what the most intense fragmentines are going to be for this blue peptide, then I can selectively extract that information out of these very, very complex MS2 spectra. And if you do that selective extraction, what you finally get back to is a signal which looks, in the end, actually very much like SRM data. And you're going to see this tomorrow yourself in Skyline. But the kind of take-home message from this slide is that the way that we achieve this continuous coverage in the mass dimension is by using this large precursor isolation window where all the blue and the gray and the red peptides are co-isolated together, co-fragmented together, and you end up with this very highly complex MS2 data. So each of these MS2 series is a multiplexed recording of essentially all the peptides across the LC, or all the peptides that are within the detection range of the instrument and are ionized. And essentially the instrument is forced to fragment all precursors in every cycle. So what we have is consistent acquisition of MS2 spectra for all analytes, again within the range of the limits of the instrument. And this, hopefully, the hope is that this will lead to reproducible quantification capabilities using this sort of user-driven extraction. And so some people find these kind of three-dimensional projections confusing, so I'm just going to try and repeat myself a little bit to make sure the point is made. So another way to visualize these types of data sets is in these two-dimensional maps, where on the x-axis we have the retention time in minutes. On the y-axis we have mass to charge, and I guess most of you have seen these kinds of maps. And each of these little black dots is essentially a peptide eluting. And so what's shown what's shown here in red is the precursor isolation scheme. So each one of these little double-headed arrows indicates the width of the precursor isolation window. And one duty cycle of the mass spectrometer is when you go from 400 all the way to 1200 with these, and in this case the window width is 25 MZ units, and it takes 32 of these to get from 400 to 1200, so we can cover the whole mass range in one cycle. And what we refer to as a swath is the sort of ensemble of all of the MS2 spectra that are acquired across the whole run using one of these precursor isolation windows for a given window. And so the duty cycle is the time it takes to get from this window in one cycle to the same window in the next cycle. So again, you can cover this mass range from 400 to 1200 if you use 32 of these windows at 25 mass to charge units each. And if you were to set the instrument up to spend 100 milliseconds on each of these MS2 scans, then you would end up with a cycle time of roughly 3 seconds, which would be important for chromatographic sampling of the peaks. If let's imagine you had a chromatographic peak width of on average sort of 30 seconds, then this would get you roughly 8 or 10 peaks across that point, and you would be able to reconstruct the chromatographic peak. So just to kind of summarize this, right? This is the 3 things that we've looked at. DDA, where you have this stochastic, semi-stochastic sampling of the precursor space, taking snapshots of these peptides, which can get to you to a lot of identifications, but is not necessarily very reproducible. In SRM, we are deterministically sampling a given set of pre-specified peptides, but we miss a lot of information that's in between these peptides in terms of the mass to charge. The precursor space is not continuously sampled. And so what we do is play a trick to continuously sample the mass to charge range by increasing the width of this precursor isolation window. But what you have to realize is that this comes at the cost of our MS2 spectra becoming much more complex, right? So we have now 4 peptides being co-fragmented, and we have fragmentines from all of these 4 peptides appearing in the MS2 scans, and we have to figure out a way to selectively extract that information out. Okay, so how do we actually extract the information, the quantitative information out? So again, here's a depiction of 3 peptides in this range now. Well, let's imagine that this is a 25 mass unit window. So for example, like 500 to 525. And we have 3 peptides in this precursor isolation window, and the width of the precursor isolation window is indicated using these gray dotted lines here. So this is an MS1 spectrum, and if we now co-isolate these 3 peptides in this wide precursor isolation window, and we fragment them, then what we're going to have is as I mentioned, a very convoluted fragmentation spectrum. So we have fragmentines from the red one, the purple one, and the yellow one, all mixed together in a single spectrum. And the question is, can we interpret this type of MS2 spectra using standard methods that are used for DIA? Yeah.
So as time goes on, the instruments are getting faster and faster.
Yeah.
So how fast do they have to be for the windows to be small enough so that your spectra resembles a DDA type of spectrum, so it's very clean? But
right.
You sample the entire space fast enough with the chromatographic time scale.
Yeah, that's a good question. That might not be too far away. I mean, this I sort of mentioned briefly in the Outlook. But if you imagine, imagine we want to use, well, let's say we want to use 2 mass unit. Let's say we want to use 1 mass unit width, and we want to cover that same mass range of 800, like 400 to 1200, for example. Well, that would be 800 scans, right? And you would need to be able to do that in the course of 2 or 3 seconds. So it would be 2 or 3 seconds divided by 800 would need to be the scan time for a single scan. So this might not be too far away. I mean, it could come in the next sort of 2 or 3 generations of mass spectrometers. And that's, yeah, I think eventually these things will converge, and then it's going to be a question of how to analyze the data.
What are the peaks that are exactly in that window?
On the border? Yeah, so what we do in that case is we put a little overlap. So it'll be full in either one or the other completely. That's the way we've handled this. This costs you a little bit of extra cycle, essentially, but that's a good way to do this.
We always measure monoisotopic peaks too, so like the whole isotope distribution still would get mangled on those edges, but we're always looking at monoisotopic fragment islands. I guess the standard fragment ions can come from isotopes of non-monostopic precursors.
Yes.
So, and I mean, it was actually discussed in the original SWATH paper, but that's sort of ignored all that.
Yeah, but we still do it. So, but there's another reason, right? So you can also score. So if you don't completely isolate the whole precursor envelope, then the distribution, the isotopic distribution in the fragment ion, so the isotopic distribution of the envelope in the fragment ion spectrum will be disrupted, right? And you can score that if you want. That's one of the scores we compute. Is like, so.
But the current SWATH methods are don't
Not for certain. Yeah, they're not wide enough. So there is but yeah, that's true. So we could make them a little bit wider, but it's probably still better than not having it. so if not having it at all, then there would be a bigger gap where you can't. The question is then, how important are these isotope distribution scores in the MS2, when it's combined with all the others? So now this gets into a lot of details, but yeah, we can come back to that. Yeah, so essentially the question here is whether these chimeric spectra are interpretable by standard kind of database searching approaches, and the answer is not really, at least not in a kind of naive way. So you might get like one of these peptides out returned to you if you were to submit this spectrum to a mascot for searching, but you probably wouldn't get all three. So the question is, what are we going to do about that? So here's a real spectrum, here's a real SWATH spectrum. And this is one of these kind of fairly complex looking MS2 spectra. And just what I want to illustrate here is basically that I know magically that this peptide is represented in this spectrum. And what I'm highlighting with these colors here, and also down here in the spectrum that you can hardly see, is the major fragment ions for this peptide. And this is just to illustrate that these fragment ions are clearly not the major, these fragment ions which we know to be the major fragment ions that should appear for this peptide are clearly not the major fragment ions in this spectrum, right? So this just shows you that you couldn't use standard methods to interpret this, and I'm just trying to illustrate the issue that, well, there's a lot more stuff in here than the peptide that we're interested in. So what can we do about that? Well, the workflow that has been developed over the last sort of 5 or 6 years, or maybe a little bit more, is this targeted analysis workflow where we take this very complex TIA data. So here's again one of these spectra that is very complicated and hard to interpret in the classical way. And we say, we turn the question around and say, instead of trying to match this spectrum in a database search, we rather say, well, is there a particular target peptide present in the data and present in the spectrum? So if we take this peptide sequence here, and we somehow have some prior knowledge that the major fragment ions for this peptide are those ones indicated in this mirror plot in pink, well, then we can kind of zoom in and say, are any of those fragment ions present? And here you see the mass range for this Y5 fragment ion, which we expect to be present. You can see this nice isotopic envelope, which is the one that would be disrupted if the precursor, if it was on the edge of the precursor quadrupole isolation window. But we can now say, okay, we're gonna look at the rest of these fragment ions, and we're gonna extract them as a function of retention time, and we're gonna plot them as extracted ion chromatograms. And so this is now the extracted ion chromatography for this peptide. These fragment ions, each of these fragment ions is plotted in a different color. And what you can see is that as a function of retention time, there's one obvious peak group where all the fragments coelute as we would expect them to, because they're all coming from the same peptide, and the peptide is just eluting once from in terms of the chromatography. So now we're back to something that looks a little bit like SRM data, and we roughly know how to interpret this. And so this is going to be the topic a little bit more of tomorrow's talk, but we can do things like we can check the coelution, whether all these transitions coelute with each other. We can check the relative fragment ion intensities versus some prior knowledge. If we know that the relative fragment ion intensities should have this pattern, we can check that the rank of these fragment ion peak areas are correlated with this. And there's various other scores that we can compute, including peak shape, and if we have reference peptides, we can compare to those as well. And this is this mProfit algorithm, which is cited down here. And these chromatography-based scores, so those of you who are in the Skyline module will have heard a lot about these kinds of things. I guess you were looking at SRM data in the Skyline module mostly. Is that right? Yes. So you will have seen a lot of this kind of data, and you'll know about these types of scores here that we can compute, and these are all based on essentially on the chromatography. So does the peptide elute at the time we expect it to? Roughly, is the cross correlation of the coelution of the different transitions? Is the cross correlation high? Is the cross correlation for the shape? Intensity is something else that we can score. Real signals tend to be more intense. Is the peak shape somewhat kind of Gaussian that we would expect for this kind of chromatography? Is there good signal to noise? And relative ion intensities with respect to a library spectrum, are they consistent? So this dot product score that you would have seen in Skyline. But now there's also spectrum-based scores. So now we have high-resolution spectra in the back. So this extracted ion chromatography is being extracted from high-resolution spectra. And so we can now ask questions about, is the whole ion series there that we would expect for that peptide? Is the mass accuracy within what we would expect for the instrument? Is the isotopic distribution for the fragment ions consistent with what we would expect? And are there interferences in these isotope envelopes? And these kind of scores in gray are only really possible with full scan types of data, whereas these blue ones can also be computed in SRM type of data. And so the software that we use to do this is called OpenSWATH, but tomorrow we're going to use Skyline, and basically it's going to do essentially the same thing. So the inputs you need are two things. You need the DIA data or SWATH data, and you need some kind of prior knowledge in the form of a spectral library. We usually do some kind of retention time alignment based on some standard peptides. And what we do is we extract ion chromatograms for the fragment ions that we expect to be there for the peptides that we're targeting. And we do some scoring to say, well, do these things, do they all coalesce? Are they consistent with the relative fragment ion intensities and so on? And then we do some statistical analysis to say whether this is indeed the peptide that we're interested in, and this part of the workflow is what, oops, all right. This part of the workflow is going to be discussed in a bit more detail tomorrow. And so just to say, this prior knowledge is a prerequisite for this type of analysis, at least in the way that we do it in routinely. And this type of information looks very much like an SRM assay. So you need to know what the protein is, you need to know the peptide sequence, you need to know the precursor mass to charge, you need to know which fragment ions you're going to monitor. So this is typically 5 or 6 fragment ions, what the charge states are for the precursor and fragments, which ions they are. Importantly, this sort of relative intensity of these ions in a library spectrum. Sorry. And also, it's very helpful to know the normalized peptide elution time. So when do we expect this peptide to elute from the chromatography? This library sort of prior knowledge part I'm going to talk about in this sort of section 2. So we're going to come back to this. Just to point out as well, though, that there are now really a large number of softwares, pipelines available for doing this type of analysis. So OpenSWATH is the one we tend to use in our lab. Skyline is what we're going to look at tomorrow, but there's many others, and they mostly fall into this categories of either peptide-centric, where we ask the question in this targeted way, is the peptide that we're interested in present in the data? Or by now there are also ways to interpret the data in a mode that's kind of more similar to classical DDA analysis, but this requires some tricks, and I'll mention these a little bit later on. But just to say that there's now a lot of options out there, and this is not an exhaustive list by any means, and it's actually increasing all the time. So I think it's instructive also to understand the nature of the data to kind of make some comparisons with SRM. So let's imagine we have SRM here. This is a triple quadrupole mass spectrometer, and we have a bunch of peptides here, and we are going to set the Q1 to a particular mass. So we're going to set it to the mass of this green peptide, and the precursor isolation width is going to be about 1 mass unit. And so we're isolating this green one, but this red one happens to be at a similar mass to charge ratio, and it sneaks through and comes along for the ride. And now we're doing fragmentation, so this is collision and this dissociation, and now we get a bunch of green fragment ions and a bunch of red fragment ions. But happily, we know what the masses of these green fragment ions are, and we're able to set our Q3 mass filter to selectively allow these fragment ions through, and so only this green guy is getting through. So we now have a selective measurement of this green peptide. And so what's plotted here is kind of a pseudo MS1 spectrum for SRM. We don't really get MS1 spectra, but this is the signal, what it would look like. And you can see that this green fragment is interference-free, so is this one. But unfortunately, this third one, the second one, has this red interference, because it just so happens that the precursor and fragment ion masses are sufficiently close to each other that this might be contaminated. What's the situation in DIA? So now I'm showing a QTOF mass spectrometer. This could also be an Orbitrap in the back end, doesn't really matter. But essentially we have the same set of peptides again, and now we're opening up this Q1 isolation window much wider, right? So here we have 1 m over Z, and here we have 25 m over Z. So that means a lot more peptides are going to get through this Q1 filter. We now have 4 here instead of these 2 up here. They're all going to get fragmented, and now we have purple and red and green and blue fragment ions, and we're going to measure the mass charge of all these fragment ions in high resolution using the TOF. And now what does the picture look like in terms of the MS2 spectra? So we still see the same green and red fragment ions, but because we now have higher resolution here, it means that we can extract the signal at a much narrower mass width, so we can do something like maybe 20 ppm. And now this red fragment ion that used to be a contamination, used to be an interference in the SRM measurement, is now outside of that extraction window, and we should have basically a clean signal, right? But it's not. It's quite as simple as that, because we also have all the other fragment ions in this picture, including the green ones from the target peptide, but also all the red and the purple and so on. And so it can be the case, of course, that one of these green fragment ions will also be contaminated. So here you can barely see there's another fragment ion here, but we can just kick that one out, right? So we don't need to keep that one. We can say, oh, there's other green fragment ions over here and here, so we can just use these essentially instead. So having this high resolution MS2 mass spectrum gives you some flexibility in terms of the fragment ion signals that you want to extract compared to SRM, which is set up, which you couldn't do in SRM because you've only measured a given set of fragment ions. What about in PRM? It's also interesting to compare to PRM. So now we have, again, this Q1 mass filter, which is narrow precursor isolation, so 1 mass unit. But now instead of a quadrupole here, we also have this high resolution time of flight, or it could be an Orbitrap instrument. So we're measuring high resolution MS2 data. And now this is actually really nice, because we have both a very selective Q1, and we also have very selective data because of the high resolution MS2. And so that means that essentially this is the most selective method out of the 3 that we've been talking about. So we have very high selectivity there, but it's not comprehensive, unlike DIA. And this is the paper came out from Ludwig Chile in our group in 2012 that kind of described this concept of using prior knowledge, so taking DIA data and combining it with some prior knowledge about the peptides that you want to dig out of that data. So what are the best fragment ion signals? What are the relative intensities of those fragment ions? What's the retention time of the peptide, and all the things I mentioned? And this is how we get back to this selective data that we came from we've developed this coming from an SRM background, so we were always trying to get back to something that's as good or almost as good as SRM data. And so using DIA in combination with this prior knowledge allowed us to do that. So that's basically the concept of this paper. And I'm going to show a few specific pieces of data from this paper. Even though this paper is not quite old, I think there's a few examples in this that are kind of instructive. So what are the possible advantages of DIA over other methods? So here's one figure from that paper. And what we have here is a dilution series of a synthetic peptide. So going from 100 femtomole, so on the x-axis here is the concentration of this peptide on the log scale. And this was spiked into some matrix, I think it was yeast, from 100 femtomole, all the way down into the attomole range. And what's plotted is basically the signal response using either SRM with the x's or MS1 quantification, which is the boxes, or using SWATH, which is the black triangles, or using or asking whether the peptide was identified or not in the DDA analysis. So this is actually the mascot score, which is plotted here, which is why it's basically flat. And what I want to point out here is that essentially, what's the order in terms of the sensitivity? Well, the least sensitive is the limit of detection for the DDA identification, which flattens out around 1 femtomole. The limit of detection for MS1 label-free quantification is somewhere a little bit below 1 femtomole. For SWATH, it's below that again. It's getting down into the attomole range. And for SRM, it's about 100 atomoles for this particular peptide. And the reason, I just show this to illustrate roughly the order which we think these sensitivities are working at. And this is, of course, just one example, but we've done this experiment now many times over the years, and we sort of roughly maintain this order, right? So SWATH is usually a bit more sensitive than MS1 label-free, but not quite as sensitive as SRM. This tends to be recapitulated in various experiments. We can also make kind of more direct comparisons with SRM experiments. So this is another measurement of about 40 or so formerly N-glycosylated peptides in plasma. So these were proteins, glycoproteins purified from plasma. And this is the SRM measurement, and this is basically the same data measured in SWATH mode. And you can see that they're very much the same, but the detail here is that we could quantify 37 of these peptides in SRM mode and only 33 in SWATH mode. So the kind of the take-home message here is that we can get close to the level of quantitative data that we had with SRM, although not quite as good. So I would say it's approaching SRM is what I usually say. And in terms of reproducibility, it's the CVs are essentially indistinguishable. So we also, at the time, took a lot of care to benchmark it versus other SRM measurements. And this is now, the biology of the experiment is not so important, but this is a classical yeast experiment called dioxic shift, where you shift the carbon source that the yeast is growing on, essentially. And what's highlighted here is the TCA cycle of yeast with a lot of the relevant metabolic proteins, and they're color-coded by, the thickness of the box indicates the copies per cell, and the color coding indicates whether these proteins were regulated in this dioxic shift experiment as measured by SRM. And then we tried to do the same experiment using SWATH, and basically we get the same, essentially the same quantification, right? So this is the fold changes plotted by SRM and the fold changes as determined by SWATH, and it essentially agrees with each other. You see these open diamonds are now the ones where there wasn't a signal in one of the so this is a heavy light experiment, I should say, and if there was no signal in the light experiment, then you don't get exactly the same fold change, but it's essentially very well correlated. And again, we could detect sort of 43 peptides by SWATH out of the 45 detected by SRM. So it's again the case where we can get almost all of the data that you can get with SRM, but not quite. What about sort of other types of added value that you might have from DIA data over SRM data? So here I'm showing now a given peptide here, and these are the transitions that you could use to quantify this peptide by SRM. So in this case, it's SWATH data, but it could also be SRM. And if you extract these 3 fragment ions, you get basically 2 peaks, right? And they both look kind of pretty good. So which one is the correct one? So if this was SRM data, you would be kind of stuck, right? It would be hard to determine which one of these peaks is the correct one. However, the point I'm trying to make with this particular figure is that if it was DIA data, you could actually just go back dynamically and extract more fragment ion signals. So if we now go back and we add for the same peptide just 3 more fragment ions, well, now we can see that all 6 of these fragment ions, all 6 of these ion chromatograms are co-eluting here, but not in this case, right? So now we can say that this is the right peptide, this is the wrong peptide, and this is something you would not really be able to do with SRM data unless you were going to go back and do additional measurements. On the other hand, you can look at it another way, which is to say you can actually remove transitions that are not working fairly easily and replace them with other ones. So here's another peptide. This is now in a light-heavy experiment, it's a N14 experiment, I think. N15 experiment. And you can see in the heavy channel that one of these fragment ions has this kind of clear contamination. Well, you could delete it if it was SRM, but then you'd be left with only two transitions. But in the case of DIA data, you can just delete one but add a new transition, right? So we delete this Y11 doubly charged guy, and we just add in this Y8, and now we get back to a clean signal. And so what I'm trying to get at here is that we have this completeness of the data, which allows you to do these kind of iterative types of reanalysis of the data and just dynamically select what you actually want to extract. And this is also illustrated again in this yeast dioxic shift sample. So initially this yeast dioxic shift experiment, they were looking at this TCA cycle and glycolysis, but then the biology collaborator said, well, wouldn't it be interesting if we could now look at electron transport chain? So again, if this was a classical kind of targeted proteomics approach, you would have to go back and do new measurements. But in the SWATH data, we could just say, okay, what are the proteins that we're interested here? And now here's all the proteins of electron transport change, and you can see that there's again changes in some of these proteins. And so this is just again to emphasize the nature of the data, the kind of complete nature of the data, that you can essentially go back and iteratively reanalyze the data. And so this is what you've seen this picture already from Rudy, but essentially what we hope we're getting here is this sort of quantitative, data quality in terms of quantification that we had with SRM, or at least approaching that, but we can now do it for several thousands of proteins, and we don't need to have we don't need to necessarily have configured SRM measurements to do this. So the summary of the DIA part is essentially, this is basically the what I pointed out to you at the start. We have fixed duty cycle, which is deterministic, and this is kind of unlike DDA, but like SRM or PRM. We have untargeted and unbiased data acquisition, which is unlike SRM, PRM, but it's kind of like DDA. We have comprehensive sampling of the precursor space, and this is the main thing to really understand in terms of how the instrument is actually being operated. And this usually means, in order to have this comprehensive sampling, this usually means that we need wide precursor isolation windows, although maybe this won't be the case in the future. And if we do have these wide precursor isolation windows, this means we have these complex mixture MS2 spectra that we need to find some way to deal with. And so in targeted analysis, or this sort of peptide-centric paradigm, based on prior knowledge, is what has helped to make DIA work, at least in our hands. So knowing something about the peptides that you want to extract and looking selectively only for those peptides. We think that the data is SRM-like in its kind of quantitative characteristics in terms of data completeness and linear dynamic range and other figures of merit like this, although it's kind of approaching SRM, as I would say. And the main advantage, I think, is that the DIA data is flexible. So you can selectively extract sets of proteins that you want in a way that kind of resembles more classical targeted proteomics like SRM. But you can also do kind of large-scale discovery-like analysis. Okay, so what about the current status? So here I'm going to show a little bit of data from So current status. So one thing I just want to mention is that the precursor isolation window scheme that I described, so that's what I'm depicting here again. So this is again 400 to 1200. And the window widths are 25 mass units each. And what's plotted here in this blue line is actually the density of peptides, or the density of peptide signals across the mass range. And you can see it's not necessarily even. So here in the kind of lower part of the mass range, we have much more signal. So a few years back, we realized it will be better to change the distribution of these window widths. So now in the most busy areas of the mass range, we have window widths which are down to like 6 mFZ, and up here around 11 to 1200, where there are not many peptides, or tryptic peptides at least, then we have 90 mass units. And this results in kind of complex samples and an increase in peptide detection of sort of maybe 15 or 20%. There are also other kinds of tricks that you can play with the precursor isolation windows, and I think maybe Mike will talk about these tomorrow in his talk, but there's other ways to get around these issues, and eventually, as you say, if the instruments get faster, this will become less necessary. And Okay, so now I'm just going to show a little bit of data from a study that we did a couple of years ago to kind of see what the status was of our ability to do this, and whether we could do it and whether other people could do it as well. So this is kind of a intra-lab benchmarking study that was conceived about, I think it was around 4 years ago. And we realized that now many labs had adopted this technology, and there was other people doing SWATH or doing DIA, and we decided we would prepare some benchmarking samples and send them around the world essentially, and ask people to measure these samples and try to determine whether the data was kind of coherent or whether it was not at all. And so this sample consisted of a complex background. This is a HEC293 lysate. And into this HEC293 lysate, we spiked a whole bunch of synthetic peptides in its kind of somewhat complicated looking dilution curve. So essentially we had 30 peptides, and we grouped them into 6 groups. And we did threefold dilution series, but we started, the starting level for each of these groups of peptides was different. So one group was started at 10,000 femtomoles on column, and it went to basically 1 femtomole on column. But the lowest one was started at 100 femtomoles in column and went down all the way to 10 attomoles in column. So essentially we covered 6 orders of dynamic range, but not with any one peptide. So this is a little bit complicated, but it's kind of a detail. And we shipped these samples out to 11 different labs around the world, and we asked them to inject these samples in a particular order. So we asked them to inject all of these 5 samples that we created with replicates of this S4 sample, and we asked them to repeat this analysis 3 times over the course of a week. And it was using relatively unsophisticated method at that stage. I mean, this 2-hour gradient with 30 centimeter columns, so not very fancy chromatography. It was also using the 5600 system, which was not the most current system at the time, but it was the one that most people had. So this is now kind of probably a 10-year-old instrument at this stage. And we asked them to use this 64 variable window width method that I just mentioned. And basically we said, okay, yeah, send us the data, and we analyzed all the data centrally and tried to figure out whether this was coherent or not. I should also mention, this is to remind me that we did have sort of an initial QC round where we sent these 5, where we sent out just the HEC293 sample, and we asked people to do a few injections of this in order to basically check the performance of the machine and troubleshoot whether there was anything wrong with the machine or not. And so here's essentially one of the main results from this study, and this is one of these curves that Rudy was showing earlier. So what's being plotted here is the number of proteins detected, and we have site 1 to site 11 on the x-axis, and within each of these blocks there's 21 injections basically. And so what you see is that everybody identifies somewhere between 4,000 to 5,000 proteins, but so the median is somewhere about 4,600 proteins. But what's important is this blue line, right? So this is the saturation curve that Rudy discussed and relies on a lot of the FDR stuff, which I'll talk about in a little bit more detail tomorrow. But this allowed us to essentially say, well, okay, if we control the FDR properly, then everybody detects essentially the same set of peptides. We don't have a lot of accumulation of false discoveries. And this is the type of data that you would want if you're going to do comparative quantification downstream, right? So you have consistent data across all the sites essentially. We can also look at these peptides which we spiked in. So these are these 30 peptides here. This is data from one site, and so again this is a dilution curve where we have on the x-axis the concentration of the peptide going from 10,000 femtomoles all the way down to 100 attomoles, and the response, the peak area essentially from the instrument. And you can see there's some spread in the y-axis dimension. This is just basically the response factor for each peptide, but essentially it's basically linear across the range. So this is the data from one site, but we can sort of draw a line, an average response through all of these peptides, and then we can overlay the data from all 11 sites. And what you can see is that it's coherent in most ways. So they basically have all this, essentially to first approximation, they have the same slope, they have the same kind of span, so there's some small differences in the limit of detection at the lowest end. There's again an offset in the y dimension, so each instrument has its kind of own response factor, but you can normalize this away without too much difficulty. But we were fairly heartened to see this result that essentially also quantitatively all the data was fairly consistent across the 11 sites. What about reproducibility? So this is the coefficient of variation, and again you have on the x-axis these 11 different sites, and the different colors indicate whether this is within 1 day. So blue is within 1 day, and light blue is not normalized, dark blue is normalized, and green is across the week. So you can see that the across the week coefficient of variation is generally a little bit higher than the within day, but we can usually normalize away using just a simple median normalization. We can normalize away most of this variance. You can see that some sites had a much higher variation across the week, but again, most of that can be normalized away. On this particular site, we could assign the reason for this was a contaminated collision cell, which is discovered by the technician after the measurement. But essentially, the coefficient variations are all below 20% and mostly below 10%. What's also interesting is this coefficient of variation. So this is the within site coefficient of variation here, these blue and green boxes. But you can also look at the coefficient of variation across the whole data set, right? So this is the CV for 229 files. And this is label-free, right? So this is all completely label-free, and just with some normalization applied. But what this gray box is depicting here is the CV if you just take all the label-free data, and it's roughly 40–50%, which is not great. If you include the normalization for the kind of response factor for each of these instruments, then you get down to somewhere where the average CV is something on the order of between 20 and 25%, which sounds kind of on the high side, but when you consider what the source of the data is, right, this is like more than 200 files from 11 different sites, label-free, we thought this was actually pretty respectable. And so something else that's interesting to look at, I think, is the difference between MS1 quantification and DIA quantification. So because we have MS1 scans in all of this DIA data, we can also extract ion chromatograms from the MS1 signals, and you'll see this tomorrow in Skyline also. And so what I'm plotting here in these curves is a percent peptide detection. So this is the completeness, what percent of these 30 synthetic peptides were detected at a given concentration, right? So again, we have the concentration on the x-axis, and you can see at the highest concentrations, basically everyone detects everything. And this is the MS1 data. This is the SWATH data, and down here is the MS1 data. And just what I'm trying to sort of point out with this is you can see that there's a difference in terms of the attrition of what peptides are detected as you walk down the dilution curve when you're looking at either MS1 data or MS2 data. So the limit of detection limit of quantification in this case is probably an order of magnitude lower in the SWATH data than it was in the MS1 data. And this is basically the summary of these data sets, right? So the orange is SWATH and blue and green is MS1 depending on how many isotopes you extract. And there's basically a gap here of 1 order of dynamic range. So the question is, what's the reason for that? And when we look into the data, here what we have shown is for 3 different peptides, extracted ion chromatography for either the MS1 data or the SWATH MS2 data. And what I'm showing here is basically at the limit of, so this is somewhere in between the limit of detection or limit of quantification for MS1 and MS2, right? So for any of these peptides, as you go down the dilution curve, you get to a point where the MS1 signal gets swamped essentially with noise from other peptides, but the MS2 signal is still pretty clean. And this has been sort of fairly consistently observed. So we always seem to get better data out of MS2 than MS1, and it will be interesting to see whether the quality of the MS1 data can be improved with newer methods and newer instrumentation that we can really fully leverage that. You can see this for yourself tomorrow in the tutorial data. One other point I thought was interesting is this. So now I'm citing a paper that came out a couple of months ago from Jesper Olsen's group, and here they did now a direct comparisons between DDA and MS1 quantification using MaxQuant and DIA using, and so this was essentially, I should say that the paper was like technical evaluation of this HFX instrument, and they did a whole bunch of different things, but one of these was a comparison between DDA and DIA directly on this sort of latest and greatest instrument from Thermo. And essentially what you can see here is that the comparison was very favorable in terms of DIA. So they got, the numbers were like almost 6,000 proteins compared to 3,800 proteins for DDA, but also the completeness was shown in these brackets is the data completeness, which was also a lot higher for the DIA data. And you can see in these histograms, the blue, dark blue is the DDA data, MS1 quantification. The red is DIA data, and then the sort of light blue is if you do a lot of fractionation, then you dig a bit deeper. And yeah, I think this is kind of interesting and a sign of the times. I hope that a group which has been very traditionally focused on kind of DDA is now coming around to the DIA way of doing things and kind of said that this looks very, very promising. Okay. What about Outlook? So now I'm coming to the end of the sort of section 1. You'll be happy to hear. So the one sort of slightly philosophical question is how to kind of analyze this DIA or SWATH data. So I said we can either do this in a very focused way where we extract kind of limited sets of peptides for just things that we're interested in terms of the biological question, or we can try to extract everything. We can extract all the peptides that we can from these large repository spectral libraries and see how this works. So this is either having a hypothesis or having no hypothesis at all, or there's many things in the middle as well, having some kind of sort of weak hypothesis or having libraries which are focused on a particular sample type. So there's certainly choices to be made here. I think that, and also, yeah, this little arrow reminds me to say that even this sort of SRM style quantification can be done kind of iteratively, which is a benefit over more classical targeted approaches. What's important to realize, and I think Rudy brought this up already, is that this kind of statistical burden associated with doing this type of analysis is higher than it is with doing this type of analysis. So as you ask more questions of the data, then the multiple hypothesis testing burden is going to go up, and so we'll discuss this in a bit more detail tomorrow. But nevertheless, I think one of the main advantages that we have in DIA data is the flexibility to be able to kind of choose between these different paradigms, and the data is essentially agnostic to what you do with it. So it's a good way of acquiring data without necessarily having to have the hypothesis in advance. And then this is the question that somebody asked already. So will the DIA and kind of DDA and also PRM eventually converge? So you can imagine a situation where we do DIA, but the precursor isolation windows eventually shrink to unit resolution, or this is equivalent to DDA sampling essentially becoming sufficiently fast that you can just cover everything, which is also equivalent to having PRM that's fast enough to comprehensively sample the entire precursor space. So then you would have some kind of super method that basically is continuous in kind of every dimension with narrow precursor isolation windows and high resolution full scan MS2, and hopefully this will be a reality soon. But then the question will be how to analyze the data. So should we do it? So you would then be able to just analyze it using standard DDA methods, database searching. But is that going to be the best way to do things? My guess is not. It will be to do this targeted data analysis, or maybe kind of hybrid approaches that combine these two types of things. But I think all the interesting questions at the moment in DIA surrounds how to do the data analysis and the signal processing and statistical analysis. And so this is I think going to be, even if the instruments get fast enough to do all this kind of thing, I think the data analysis is still going to be where the rubber meets the road. All right, so I'm going to pause here for a minute. Are there any questions at this stage?
So when you mentioned calculation of the species, you said the data was not unnormalized and normalized.
Yeah.
What do you mean by normalization?
So in this case, so remember in these samples we had basically a HEC293 background and we had synthetic peptides spiked in, right? And the data, the quantitative data that I was showing in those dilution curves was for the synthetic peptides. So the normalization that we were doing in this case was just to take all of the peptides that we could extract from HEC293 and just do a median normalization. So we were using the background to normalize the synthetic peptides. And one of the advantages of DIA potentially over methods like SRM is you have a lot of features, potentially thousands or tens of thousands of features to do different kinds of global normalization if you want to. Yeah. Yeah, that's a problem. So yes, exactly. I mean, the short answer is no, I think. We can try some tricks to avoid this problem. So I think that you're right that we can have different, so the question is, I should stand over here next to the microphone. The question is, if we have such a wide precursor isolation window, we can have different precursors in there that have different charge states, and we can only apply one collision energy to this precursor isolation. So should it be the one for the doubly charged or the triply charged or the quadruply charged? And if so what we do actually is we just choose the collision energy for doubly charged. This likely means that the triply charged ones are being over-fragmented, or the higher charge state ones are being over-fragmented. And that's going to degrade the quality of that data, and actually we've now seen some evidence of this. One thing you can do to try and offset this is do a ramped collision energy, basically. And we do that. We go from sort of minus 15 volts to plus 15 volts in the hope of kind of, at some point, catching the right collision energy. But yeah, I agree, this is an issue. And it's not totally clear to me that it's one that can be solved easily. Yeah.
Looking at the reproducibility side of the data, it said that site B has technical product.
Yeah.
So if you remove variable A because the this is outside of the will you see across all sites efficiency for that?
For the most part, yeah. I mean, there are some sites which are sort of in between, right? You see like site 2 or 3 also had some deviation across the week. So certainly all of the normalized values, they're definitely all below 20% and mostly below 10%. So the normalization in label-free analysis is definitely worth doing. And the normalization that we applied is fairly naive, right? If this is just median normalization, there's probably cleverer ways to do that. I should also say that the way that we selected, well, this is now peptide level, but we also did protein level CVs. I didn't show that. And the way we selected the peptides to infer those proteins and to quantify those proteins was also fairly naive. We just did some kind of top 3, which is not the smartest way to do it, right? There's certainly smarter ways to select which peptides you want to use to do protein quantification. I think that's also going to come up tomorrow. So I think that there are tricks that can be played in the data to improve this too. But yeah, I think the short answer is yes. Yeah. Yeah, so I mean, there's no magic happening there. So the dynamic range that we can achieve is something like we see from these dilution curves, something like 4.5 orders of magnitude, and this is maybe pretty much in line with SRM. SRM maybe you can get 5 or a little bit more. So in plasma, for example, we've done studies in plasma. This translates to roughly 400 proteins, something like this, that you can detect when you don't do any depletion, you don't do any enrichment. So I'm not sure that, yeah, I'm not sure there's much more to say about that. I mean, we're limited by the dynamic range of the instrument, and it's going to take work from the vendors to increase that dynamic range primarily. That's not the case, actually. So my experience is mostly with QTOF instrumentation. If you now go to trapping instrumentation like quadrupolar Orbitrap, there I think you potentially, in those kinds of more simplified sample matrices, you might see some benefit in terms of dynamic range because of the ability of the instrument to basically accumulate more ions. And so I think Mike will talk tomorrow about doing DIA on trapping instruments more specifically. So I think in complex samples, the difference between the machines might not be that much, but for samples where you have low amounts of sample material or you have low complexity in the matrix, then you might get better sensitivity and more dynamic range using trapping instruments. don't know if did I answer the question there, or okay, we can talk about it later also if I didn't. Yeah.
So I have a question about if I add some modification once the phosphorylation in the data set, it will give a lot of false positive because the mixture spectrum makes it very complicated.
Yes. Well, so, I mean, there's a couple of layers to that. We have recently, so certainly the way that you score PTM peptides is going to need more attention than you do for kind of unmodified peptides. So particularly like isobaric modifications like phospho, where it could be here or there, and you need to look at diagnostic fragment ions to localize that. And we just published a paper about half a year ago, I guess, called, we published an extension of our standard open swath tool called IPF, Inference of Peptidoforms. And basically what this does is a two-pass analysis. So it first tries to find like high quality signals that look like the peptides that they should be in the normal way. And then it re-extracts all of the fragment ions that could be useful for localizing particular isoform modifications, isobaric modifications, and sort of overlays these onto the peak group and then scores these using its Bayesian hierarchical model, and gives you some kind of metric about whether the localization is here or there, or whether it can't be, there isn't enough evidence to say. So it's really only, and there's also a tool from the Macass lab that does something similar, which is not quite out yet, but I think is going to be out soon. So the tools are coming online to do that, and they have decoy models which account for the problem that you are describing, that the spectra basically are more noisy. So that should be accounted for in those models. I guess that's what I would say about that. I would recommend looking at this IPF paper that came out in Nature Biotech in the last year or so. There's a lot of detail about that. It's a whole big topic. Yeah. Yes.
I have a couple questions. Is there a public library available with spectral libraries?
Oh, so that's what I'm going to talk about next in much more detail. Yeah.
The other one was, I mean, you can't apply eye track to these, so how do you compare, how do you quantify it? And we're going to talk about it.
Yeah. So most of the time we do label-free quantification, which means we do one injection of each sample, and we compare the peak areas from the first injection to the second injection and make a ratio or something like this. And then, so you're right, we definitely can't do eye track because the isolation windows are so large. It's the opposite of what you want with eye track, right? With eye track you want to have very well isolated precursors. So, I mean, DIA is also compatible with doing other kinds of isotopic labeling, either synthetic peptides or SILAC or other kinds of metabolic labeling. These can be included, but they do need to be handled properly in the software. And we have seen, because especially in the case, for example, of SILAC, when you try to analyze a SILAC sample, sometimes the sensitivity goes down, the number of peptides you can detect goes down, because what you're doing is taking very, very complex spectra already and making them twice as complex, basically. So while you can do SILAC and it does benefit you in terms of the quantification potentially, it can be a hindrance in terms of selectivity and being able to actually selectively quantify the peptides you want. So, I mean, this is a long way of saying that most of the quantification we do in our lab is done in label-free mode. Yeah.
So you mentioned there were a bunch of different softwares used for the automated analysis
Right.
of this data. Is there like a particular software for certain things you're using specific software, or is this kind of like a whole bunch of different software that use pretty much the same thing?
Yeah. It's kind of a mixture of those things. So we have a mixture of kind of academic software, which is either, for example, the pipeline from our group called OpenSwath is a good pipeline, but it's not particularly user-friendly. So I wouldn't necessarily, if you're sort of brand new to this, and you don't have computational support, I wouldn't necessarily go down that route because it's a lot of annoying kind of command line. It's good for us because we can automate it, it's scriptable, we can run it on a cluster. It's a very good place to develop new algorithms. You can do fast algorithm development there. So for our purposes, it works very well. If you want to go for something more user-friendly, then I think Skyline is a very good option, and you'll see that tomorrow. But Skyline is also a software that tries to do many things, right? It does SRM, it does PRM, it does MS1 quantification, it does DIA, it does peptides, it does small molecules, it does a lot of things. So Brendan can't necessarily keep up with every cutting-edge paper that comes out. So he's very good at adopting the best stuff into the software and trying to keep that up to date, but it doesn't necessarily, sometimes it takes a little bit of time. Then you can go to like commercial implementations. So there's Biognosis, this company Rudy mentioned, also produced a very kind of slick software which is very well optimized, runs very efficiently, is easy to use, is probably reasonably well supported, but then you got to pay some money for this, obviously. So I would say there's a range of options. Also now Scaffold, some of you may be familiar with Scaffold, they have a DIA option as well, which I expect will also be nice and commercial and easy to use. So there's a kind of a range of purposes for these softwares, depending on whether you're doing kind of algorithm development or really you just want something that works. So I think Skyline is a good place to start, and you'll see tomorrow how this operates, but there are certainly a lot of options out there. Other questions now? Okay, so we've got, I'm supposed to finish at 5, right? Okay, so we don't have that much time left. I'm going to quickly go through, so this is sort of part 2, spectral library generation and public library resources, and I just want to mention some details of this, and we'll try to get through the first maybe third of this and try to cover some more of it tomorrow. So the outline here is why do we care about prior knowledge in targeted proteomics or in DIA in this case. The second part is I would like to just kind of quickly walk through the procedure for generating spectral libraries and these what we call peptide query parameters, right? So the coordinates for extracting the data, and point out a few issues here. And you'll see some of this in Skyline tomorrow. And then the last topic will be the one the question was about, is whether there's publicly available spectral library resources, and do you actually want to use these? And the answer is sometimes yes and sometimes no, I would say. But let's start at the start. So why do we care about prior knowledge? Well, the first thing you want to do is to be able to direct the software which signals to extract from the data. So which fragment ions from which peptides are likely to be the good fragment ions from the best peptides. And the second part is the prior knowledge can also be used to score the measured data against. So I mentioned in the first talk that we can look at the relative fragment ion intensities in the library spectrum, and we can compare this to what we see in the extracted data and see whether they're coherent. And we also can know something about the chromatographic elution. So that means we know where this peptide should appear in retention time. And so this is useful information to have. And so, I mean, I showed this slide already, but just to remind you what the problem is, right? The problem is we can't extract, we can't use standard methods to search these data, so we need to do some kind of selective extraction. And so a question we used to hear a lot would be like, well, we would go to meetings and say, oh, you can do DIA and SWATH, and you just need this prior knowledge in terms of a spectral library, and if you use it to do this kind of peptide-centric data analysis or targeted data analysis, well, then you can get complete and sensitive or robust data out. And people would say, oh, yeah, but where do you get these spectral libraries? This was sort of not really clear to people. I think it was people who had done some kind of targeted proteomics understood this, but people from the DDA world or from outside were like, didn't really get this idea. So what we did a couple of years ago was to write this protocol paper that describes in agonizing detail how to make them. And what I would usually do now is to kind of go through this procedure. And so I'll try to point out from a high level what some of the important features are, although we're now running out of time, but we can finish the rest of it tomorrow. But anyway, if you're interested in this topic, I would encourage you to take a look at this protocol. And the goal here for us was really to kind of document what we thought was the best practice in terms of these libraries at the time, although things have probably moved on a little bit by now. And also to kind of document, if we were going to put out spectral libraries into the public, we would be able to sort of document how these were made. And so again, the type of information that's being contained in these spectral libraries are actually in what we would call sort of peptide query parameters, right? So the set of coordinates that you would extract from the DIA data is what I mentioned already. It kind of looks like an SRM assay. So it's the peptide, the protein, the sequence, the masses of the precursor and fragmentines, the charge states, which fragmentines we're actually looking at, the fragmentine intensities, and the normalized retention time. And so the question is, how do we get to these? So I've only got 8 minutes left, so this might be I actually think this might be a good place to stop until tomorrow.